Here we have a close-up look at the structure of a double-stranded DNA molecule. So a few things to point out here is notice that the 5' end of one strand is going to be across from the 3' end of the other. So that's that anti-parallel nature that we would talk about. And then also notice that on the outsides, so here and then also over here, you have, if we just trace it down like this, we have sugar, phosphate, the whole way down. So we do tend to think of the outside of the helix as alternating sugar phosphates. And that leaves on the inside of the helix all of the bases. So notice in the inside, in the interior, that is where the bases would be found. Now here we're really looking at a very, very short piece of DNA. We would assume that this molecule would continue to go on and on because DNA molecules are very long molecules. But if we look closer at the bases and we talk more about these base pairs, they are shown as little dotted lines here, but if we just highlight them a little bit more, these would be all of the hydrogen bonds that you do find between the bases and really what holds the two strands together. And notice that between A and T, we are going to get 2 hydrogen bonds, and between G and C, we always get 3 hydrogen bonds. And that will always be the case when we find these inside the DNA double helix. Notice also within the base pairs that it's always going to be a purine, and so here would be our purines. It's always going to be a purine across from a pyrimidine. So cytosine and thymine, those are the pyrimidines. So what this means is that you always have a large base across from a smaller base, and this will help us to maintain a consistent width down the DNA molecule. Now, before they really knew the structure of the DNA molecule, an individual named Edwin Chargaff, he looked a lot at the base composition of DNA. So in other words, he studied what the percentages of the various bases were in the DNA of different organisms. And he came up with what we call Chargaffs Rules. And so Chargaffs Rules basically stated that the percent of adenine, or A, that you find within a species is going to be equal to the percent of thymine. And he also said that the percent of guanine is always going to be equal to the percent of cytosine there. Now, at the time, he didn't know really the reason for that. He just knew that those rules always held true if you were looking at the composition or the percentage of the bases in a particular organism. So these do hold true, we now know, because those are the base pair combinations. So where you have an adenine, you will always find, if you look inside the helix, you'll find those two across from each other. And the C and G will always be across from each other as well. Well, the usefulness of his rules is that if we know one of these bases, what its percentage is, we can predict the base percentages of the other ones. So for example, if we looked at an organism and we found that the percentage of A was equal to 15%, then we could take that information and we could find the percent of T, the percent of C, and then also the percent of G. So the way that we would do that is, again, if we look over here and we know that the A's and the T's, those percentages are always equal, that makes it very easy to figure out what the percentage of T must be. And so it's also going to be 15%. And then if we consider that all 4 of these have to add up to 100%, well, here we have 30. So between these two, we must have another 70% that we would want to divide up. So then we just take 70% and we divide that by 2, and that's going to leave us 35% for C and 35% for G. And then we could double check and just make sure again that the C and the G, they are equal to each other. And then again, if we added up all of them, that they are going to equal 100%. So anytime you're presented with one of the percentages that we have of a nitrogenous base within a double-stranded DNA molecule, we should be able to figure out what the percentage is of the other 3 bases.